Good morning, everybody. So I'm going to present you over the next 15 minutes the activity of PDC line Pharma, who is a company involved in cancer vaccine based on a very specific off-the-shelf antigen-presenting cell line. In terms of company profile, we're a company of 27, 30 now, close to 30 people, based in Belgium and France. In terms of funding, we raised close to 52 million euros so far in equity and non-dilutive money. The technology is in fact coming from the French Blood Bank, which is a large organization in France that collects the blood, but as well as academic research lab associated. And the scientific founder of the company was leading one of his labs, and he has identified in the blood of leukemia patient a very rare leukemia affecting the plasmacytoid dendritic cells. In the dendritic, in the blood, you have majority of the cells that are the myeloid dendritic and a small fractions that are the plasmacytoid. And unfortunately for the patients, the mutations and the disease was affecting the leukemia, these small fractions. But from the blood of his patients, he developed, scientific founder, a cell line, immortalized cell line, that we are still using today as a vaccinal platform. And I will come back to explain you how it works. So the technology has three key advantages. It's immortalized cell line, so leukemia, so it can grow in culture and suspension, so it's very easy to expand. The origin of the cells being plasmacytoid provide very interesting property in terms of capacity to prime and expand cytotoxic CD8 T cells, and it's a versatile platform. In fact, we just have to load the cell line with GMP peptide, representative the tumor antigens we are selecting, and we can, in fact, address any kind of solid or liquid tumors. But today we are in clinic in the lung, non-small cell lung cancer. A first feasibility academic study, first in human, has been done in melanoma, but the company is now really focusing on non-small cell lung cancer. Important to mention, we already signed a first licensing deal before starting, in fact, the clinical trial in the lung, which is ongoing now, with LG Chem. So LG, I'm sure all of you knows for this flat screen, the TV, electronics, but it's as well the 10th chemical company in the world, and we have an increasing growing life science department in Asia, and we license out the lung cancer vaccine for Asia. They have straight license for South Korea and option for the rest of Asia. So at the top of the money we get, it was important to have a partnership, and because they will co-develop the product in Asia and at the end commercialize the product in Asia. Important assets at the top of the technology of the company is the management team. So we have very experienced people. For myself, already co-founded several biotech companies. I've been involved in IPO, M&As. The medical director, in fact, led the major cancer vaccine trial of GSK vaccine and in lung and melanoma. So she has exactly the right profile for us. So she joined the company full time. Claude Dédry, the head of the head of the CMC, has a long experience in cell and gene therapy, and he's a former GSK people. The scientific founder joined full-time the company, and the CEO, CFO has as well a good experience in oncology. In the board, we have as well very experienced people. I would just to mention the arrival of Monder Majoubi 2 years ago, who is the former executive VP of Genentech Oncology, who is the president of our board. He's of course an oncologist. He's the current CEO of Pharma. Jean-Paul Prils, who is the former global R&D director of GSK Vaccine. He has invested personally in the company, so it's as well a mark of trust. Alain Herrera, the former head of Sanofi Oncology, and of course representative of the investors. I will come back later, but you see one of the key investors of the company is Korea Investment Partner. You know, it's a big South Korean fund. They have more than 3 billion investment. And we have, in fact, in the capital of the company, more than 5 South Korean funds. That is related to the deal we signed with LG Chem that provide us awareness and credibility in the ecosystem and access to this funding. So today, in terms of financing, we have a mix of Belgium and South Korean investors. So the company made significant achievement over the last 24 months. I just want to stress some of them. We raised 2 rounds of financing, B1 and B2, to finance the clinical trial, which is ongoing. 20 million B round in 2019 and 17.5 in November 2021, both led by Korea investment partner I mentioned earlier. In February 2020, when the first patient with non-small cell lung cancer low dose, we had the arrival of Monder Majoubi in June 2020, the new president. We are testing 2 doses of vaccine, low dose and high dose. The high dose was administered to the first patient in September. We get in December 2020 100% compliance of the 6th batch of the lung manufacturer, so it's extremely important in our field to have the CMC under control, and we can see at the scale we are today, it's completely under control. We opened the last cohort of patients, which is in fact high dose in add-on with checkpoint inhibitor, because we are targeting an activity in associations of our vaccine. And it has been opened in September, and as I said, we closed the B2 round of financing in November last year. Okay, how the technology works. So we have a master cell bank, where the cell line from the leukemia patients I mentioned earlier has been expanded, purified, characterized, and stored in liquid nitrogens. It has been approved for clinical use by the French authorities. And from this master cell bank, we have developed working cell banks, to expand the capacity. And when we want to launch clinical batches, what we have to do is just to seed a single-use bioreactor. We have today 1 liter and 10 liters bioreactor. The cells grow in suspension in synthetic medium. We are working with the Lonza serum-free medium. And after 9 days of culture, we have billion and billion, more than 20 billion cells. Then at the harvest, we split the cells according to the number of peptide we want to load on the HLA present in the membrane of the cells. We target HLA-A2 because the patient is a Caucasian, was HLA-A2 positive. And after we pull the cells that are loaded with a peptide, irradiate to stop the proliferations, it's leukemia cells. Then we put in the technology of aseptic technology and with a cryoprotectant, it's a vial resistant to cryopreservations. And like for the COVID-19 vaccine, we ship the cells in minus 80 degrees to the hospitals, put in fridge, 80 degrees fridge. And when the patient is ready, we thaw and infuse the vaccine under the subcutaneously or IV. And the vaccine will go to the lymph node, prime, expand CD8 T cell that we migrate to the tumor microenvironment to make the job, which is to kill the tumor. In terms of potency, we did several benchmark studies to compare the ability of our cell line to expand cytotoxic CD8 T cells. And we've taken 1 antigen, which is the melan, and we compare with other approach, which are the conventional autologous dendritic cells or allogenic dendritic cells that are both myeloid and DC. And as you can see here, in vitro by fax dextramer, we are 20 to 200-fold more potent than this classical approach, which is at the top of the manufacturing and the cost of good and the consistency of our manufacturing key advantages for technology. And that is related to a very specific mechanism of actions, but I don't have to go in details today, but I can answer to the question later if you want. We did study as well with neoantigens. It's a hot topic currently, and for that we have selected 2 neoepitopes. So with mutated peptide, where the amino acid mutated in red has been clearly identified and characterized and published in the literature, identified in the blood of melanoma and lung cancer patients, and put in culture with purified CD8 T cell of 3 healthy donor, what you see here, and that is the output of the fax. In red and purple, and as you can see with the cell line, we can definitely expand neoantigen as effectively that shared antigens, so it works for both, knowing that in a healthy donor, the frequency of these naive CD8 T cells, because of the cells are naive, is extremely low, 1 out of 1 million. So once again, showing the potency of our cell line, the ability to expand very rare population of CD8 T cells. Okay, we have demonstrated and published as well that our vaccine is can be boosted by the checkpoint inhibitor, the combinations of the vaccine activity. You know, the checkpoint inhibitor block the mechanism of defense of the tumor, and the vaccine increase the immunity. So it's something we are looking for to get this synergetic effect. So you see here in vitro, we've taken the blood of melanoma patients. You know, melanoma is a very hot tumor with a very high tumor-induced immunosuppression. And you see here, after 1 week of culture, we can have 15-fold increase the vaccine alone. With the checkpoint inhibitor alone, we have almost nothing in terms of expansions. But when you combine both, the vaccine and the checkpoint inhibitor, is when you see this very nice synergetic effect between the vaccine and the checkpoint. And that has been done on a cohort of 13 melanoma patients. So today we are, as I said, targeting HLA-A2 peptide. We are using HLA-A2 restricted peptide, but we have genetically modified the cell line to express additional HLA, like the A24 or B35, which is to increase the coverage in terms of patient population, and it works very well. Today we are loading the cell line with peptide, GMP peptide, because it's the easiest approach. It's off-the-shelf for technology, so we have time to produce and store the vaccine. We don't have time limitations. We tested as well retroviral transductions to have endogenous expressions and presentation of the peptide by the HLA by the cell line. It works well as well. And we did as well electroporation through mRNA, and we had as well good results. So according to the indications and to the market we are targeting, we can adjust the tumor antigen loading strategy. Okay, let's finish by the lung cancer trial, which is the clinical trial ongoing today. You have here the 6 antigens we have selected for the vaccine. The vaccine is loaded with these different lung-specific antigens with a good coverage in non-small cell lung cancer. You have MAGE-3, MAGE-4, MUC1, New York ESO1, Survivin, and Multimage. And as you can see, in vitro with a healthy donor, we can definitely expand the CD8 T cell for all of these antigens. It was a primary assessment we did. The current clinical trial design is presented here. The objective is to assess safety, immunological activity, and clinical response of the vaccine in combo with checkpoint inhibitor. We are testing 2 different doses, low dose and high dose. First with the vaccine alone in monotherapy. It's in adjuvant setting, so it's phase 3A patients. So where after surgery and in general adjuvant chemotherapy in a washout period, we infuse 6 times the vaccine. And we have 6 patients low dose and 10 patients high dose. And after we have the second population, which is the targeted for the clinical benefit, first line metastatic patient in combo with checkpoint inhibitor, low dose 6 patients and high dose 42 patients. Okay, and we have already completed the 3 first cohort, and we are still enrolling on the last cohort. We are infusing IV and subcut weekly 6 times. Okay, to close my presentations, just the line of time and the funding strategy. So we expect to get the final result of the clinical study I presented just before, Q1 2024. We should have an interim report mid-2023 based on the 19 patients of the B2 cohort, the last cohort. And we are still ongoing on the maximizing the value of the current technology in terms of characterizing the mechanism of actions, why this cell line is so potent, neoantigens and the CMC development. We would like to go to up to 50 liters bioreactors to move from 1, 10, and 50 liters. We will have our GMP facility established next year. And as I said, we get 2 rounds of financing already, and we expect to do a new round of financing a few months after the result of our clinical study report, and to be able to finance a second clinical trial that we expect to be a randomized proof of concept study that will be able to demonstrate the clinical benefit of our product. Of course, we will carry on. I didn't show you the pipeline, but we have other indication in mind, and we have as well activity in immuno in neoantigens, and we'd like to set up in US, in Asia, from 2024. And this is the end of my presentations, and I would like to thank you for your attention. Thank you.